Hello, and welcome to the Drug Discovery World podcast, a podcast covering topics around drug discovery and development, pharma, and biotech. My name is Giles, and I'm here to take you through this episode. Today's episode is taken from our current summer 2020 issue, and is part 2 of 2 episodes, titled Race to COVID-19 Cure, the core model for solving the pandemic crisis. This article was written by Ibis Sanchez-Serrano and Rathnam Chaguturu, and will be giving their full author bios at the end of this episode. So now onto the main article. Race to COVID-19 Cure, the core model for solving the pandemic crisis, Part 2. The United States government's Operation Warp Speed program has selected 5 vaccine projects: Moderna, Oxford University and AstraZeneca, Johnson & Johnson, Merck, and Pfizer, to receive billions of dollars in federal funding and support, again, before there is proof that the vaccines work. Vaccine development is time-consuming and financially exhaustive, attrition is high, and it is often an exercise in futility. Effective innovative paradigms are needed, but never forgetting safety over speed. Even if a vaccine is developed, several questions remain to be addressed. Do people develop immunity? If yes, what kind of response and how long does it take or last? Although the Society of Critical Care Medicine cautions against treatments using drugs, and the Infectious Diseases Society of America recommends patients should get the drugs only as part of a clinical trial, treatment options offer a viable promise, as has been the case against HIV. Drug repurposing or drug repositioning, the identification of new indications from existing drugs, has in recent years successfully identified promising candidate drugs that can open new therapeutic avenues and offers great promise to counteract COVID-19. Several companies, AbbVie, Amgen, Bayer, Gilead Sciences, Incyte, Eli Lilly, Mesoblast, Novartis, Regeneron, Roche, Sanofi, Takeda, etc., have approved products on the market that could hold potential in treating patients with COVID-19. Others have experimental drugs included in testing for other viruses that could be effective in targeting novel coronavirus infection. For news concerning COVID-19 therapeutics development, refer to Coronavirus Today at coronavirustoday.com/COVID-19-treatments. Drug therapy, either singly or in combination, for example, remdesivir, chloroquine and/or hydroxychloroquine, baricitinib, Acterma, convalescent plasma, ivermectin, Kaletra, etc. for COVID-19, has a dual purpose of both preventing the virus from spreading inside the human body, as well as protecting the body's immune system from the havoc caused by the viral attack. The FDA recently created a new emergency program, the Coronavirus Treatment Acceleration Program, aimed at speeding up research for the development of COVID-19 treatments. The investigational antiviral drug, remdesivir, Gilead Sciences, received Emergency Use Authorization, EUA, by the FDA on May 1st, as it appears to help some hospitalized SARS-CoV-2 patients to recover faster. In Japan, at the discretion of the Health, Labour and Welfare Ministry, early-stage patients may receive the antiviral drug Avigan. The most pressing, immediate need in the battle against COVID-19 is for diagnostic tests to determine if individuals are infected by the novel coronavirus and post-treatment follow-up. On January 7, 2020, Chinese authorities isolated and identified the new coronavirus, whose genetic sequence was shared internationally on January 12th, so that countries could use it to develop specific diagnostic kits. In our fight against COVID-19, as with any epidemic, testing, and more explicitly, the capacity to perform tests for a large sector of the population, is fundamental. Massive testing not only allows the healthcare system to identify, monitor, and treat people who have been infected by the virus, but also to trace their contacts, who may be potentially infected, to detect foci of infections, and access the healthcare, logistic, and economic resources available, and the ones that are needed. The better our knowledge of the actual number of infected people is, the better our decisions to mitigate the disease. A given number of tests at the national level daily could provide valuable insight on the basic reproduction number, R0, and guide key decisions such as the adequate timing to lift lockdown restrictions, and the way in which this should be accomplished, the reopening of the economy, active surveillance to avoid potential second waves, and when to end lockdowns completely. At the individual level, testing can guide the treatment of a given patient, as well as determine when that patient can be cleared and/or whether he or she has developed immunity, something which in the case of SARS-CoV-2 is at this point very uncertain. Immediately after the Chinese researchers published the genomic sequence of SARS-CoV-2, a number of companies became engaged in activities related to testing for SARS-CoV-2. These include, among others, Abbott Labs, Becton Dickinson, Co-Diagnostics, Danaher, LabCorp, Quest Diagnostics, and Thermo Fisher Scientific. Since then, a number of tests have been developed. The testing strategies include methods that detect the presence of the virus itself, RT-PCR, isothermal nucleic acid amplification, antigen, and those that detect antibodies produced in response to infection. These tests detect the presence of SARS-CoV-2 virus, for instance, reverse transcriptase polymerase chain reaction, and its variants, isothermal gene amplification, and antigen test. They can detect the antibodies produced in response to a previous infection, for instance, serological tests for the detection of immunoglobulin M, IgM, and immunoglobulin G, IgG, specific to SARS-CoV-2. And finally, they can assess the damage that SARS-CoV-2 is creating in the organism, especially in the lungs and in the brain, for instance, computerized tomography scan, CT scan, and oxygen take-up test. One of the major challenges for early detection of SARS-CoV-2 has been what type of test is more reliable. For instance, RT-PCR could produce false negatives depending upon at what stage the sample is taken. In addition, it is relatively time-consuming and requires well-trained personnel, and it is not appropriate for fast mass testing. Isothermal nucleic acid amplification-based tests are fast. They can provide results in 13 minutes, but not sufficiently reliable. Antigen tests are cheaper and simpler than the RT-PCR test, and can provide readings faster, 15 minutes, but are less accurate, as a positive case may come up as a negative because the level of antigen in the sample is below the detection limit of the test. So, an RT-PCR test may need to be used to confirm or discard a suspicion. Thus, there is at present a race by many companies to come up with test kits that are cheap, fast, and reliable, just like a pregnancy test. So far, the United States FDA has granted clearance for emergency use to 3 test kits, in which the patient, under prescription, can take a nasopharyngeal swab or saliva sample at home and then send it to a diagnostic lab. And many other countries, in all continents, are doing the same. With COVID-19 infection varying in severity from asymptomatic to lethal, the search is now underway for specific biomarkers as clinical predictors of mortality and morbidity due to COVID-19, and help speed up clinical trials and reduce the cost of drug development. According to Global Data's biomarkers database, the top 2 biomarkers being utilized for COVID-19 trials are diagnostic markers. The first is severe acute respiratory syndrome coronavirus 2, SARS-CoV-2, per se, which is currently being used in approximately 30% of COVID-19 trials. And the second is coronavirus nucleic acid, which is being included in just over 5% of trials. In COVID-19, the severe acute respiratory syndrome appears to result from a dysregulated hyperinflammatory response manifested by the release of excessive pro-inflammatory cytokines and chemokines, coined a cytokine storm. The main cytokines appear to be IL-6 and LIGHT, homologous to lymphotoxin, exhibits inducible expression, and competes with HSV glycoprotein D for binding to herpes virus entry mediator, a receptor expressed on T lymphocyte. Other biomarkers used in COVID-19 trials include C-reactive protein, lymphocytes, and SARS-CoV-2 RNA, procalcitonin, and SUPAR. In addition to the work being carried out by biopharmaceutical companies, research institutes, and academic centers, several important initiatives are being performed at multiple levels. For example, the WHO has been leading the international efforts to control the pandemic. It has activated its R&D Blueprint initiative to accelerate diagnostics, vaccines, and therapeutics for this novel coronavirus, and sent more than 2 million personal protection equipment, PPE, and testing kits to more than 120 countries. It has also launched the Solidarity trial, aimed at assessing the most promising treatments, among many other initiatives. The FDA has created a special emergency program for possible therapies, the Coronavirus Treatment Acceleration Program, CTAP, as well as many other strategies. The UK has launched its Recovery trial, which is the world's largest COVID-19 one, with more than 5,000 patients already taking part. The National Institutes of Health, NIH, among many different initiatives, has recently launched the Public-Private Partnership for Accelerating COVID-19 Therapeutic Interventions and Vaccines, ACTIVE. Big pharma companies are trying to develop treatments and vaccines through alliances and a collaboration with academic research centres, as discussed previously, while multiple research centres around the world are exploring the use of blood, plasma with antibodies, from patients who have survived COVID-19 as a treatment. Also, philanthropic organisations such as the Bill and Melinda Gates Foundation and the Wellcome Trust, among many others around the planet, have poured in funding to fight COVID-19. In addition to this, and to what every country is investing in their own national fight against COVID-19, recently, world leaders from France, Germany, the UK, Japan, European Union, Italy and Spain, and other countries pledged €7.4 billion that will be channeled through global health organisations such as CEPI, Gavi, the Vaccines Alliance, the Global Fund, and UNITAID, for research to find a vaccine against the new coronavirus. The severity of the COVID-19 pandemic has prompted bioethicist Nir Eyal and epidemiologists Mark Lipsitch and Peter Smith to propose carrying challenge trials that might ordinarily be dismissed as too risky. The proposed challenge trials deliberately expose low-risk, healthy volunteers to SARS-CoV-2 to quickly learn more about the disease it causes, or to gain early insight into whether a new vaccine or treatment works, thus speeding up the development of a vaccine for SARS-CoV-2, reducing the global burden of COVID-19 related deaths, and potentially saving millions of lives. Geneva-based SGS and London-based hVivo are gearing up to carry out the challenge trials in coming months. More than 28,000 volunteers in 102 countries have already signed up to participate in these challenge trials. Beyond ethical and legal issues, there are significant logistical challenges to conducting such trials, and high stakes of exposing volunteers to potentially fatal risks. It would be a mistake to rush the implementation of challenge trials without adequately considering what it will take for them to make a difference. All of this, vaccine development, COVID-19 treatment, and diagnostic testing, requires a strategy to integrate prevention and treatment options as one continuum, parallel over traditional sequential paradigm. With the diversity in the ever-expanding operational models, a proven strategy is needed that is flexible to accommodate and address changing demands of the partners, and should be the ultimate choice for vaccine and drug discovery and development. Until proven prevention and/or treatment options emerge, keeping the SARS-CoV-2 contained is crucial, requiring aggressive testing, tracking, and isolating contacts of people who have the virus, and continued physical distancing. The pandemic has exposed cascading weaknesses of varying proportions in global disaster response among countries and healthcare agencies within a country. What once worked is no longer applicable, as evidenced by the long-standing procedures for sheltering victims in stadiums, churches, or other crowded spaces suddenly become dangerous because they risk worsening the pandemic. According to Chris Curry, United States Government Accountability Office, the COVID-19 pandemic has complicated every aspect of disaster planning and response in a way that we have never experienced before. In combating the COVID-19 pandemic, we need to be reminded that we win or lose together. Now is precisely the time for a pragmatic, operational model that integrates and swiftly addresses every aspect of the pandemic response, from diagnostics to vaccine development to treatment options. It is clear that many sectors in society are willing to cooperate and join forces. But there is a fundamental issue acting against this. All these alliances and efforts are scattered and unsynchronized. there is not a coherent and focused operational organisation in them. If these forces were joined cohesively through an organisational collaborative model, a cure for COVID-19 is possible sooner than expected. This is why the implementation of the core model, which we'll now discuss, can become crucial to combating COVID-19. The core model. The core model is an organisational model for research and development and an economic theory. It lays out an adaptable, novel organisational and design paradigm for drug discovery and development based on collaboration. It is also an economic theory for innovation and economic growth. The core model consists of three main structures, named core, bridge, and periphery, through which knowledge is transferred, integrated, and finally translated into commercial products, via an orchestrated exchange or trade of assets, exchange of materials, animal models, knowledge, data, clinical trials results, personal connections, resources, funding, etc., among players and structures, which forms the basis of collaboration. This strategy allows for bi- and multi-directional interactions that significantly save time, effort, and resources, and lowers the amount of energy necessary to reach the agreed-upon common goal, from a high state to a low state. An important finding in the core model is that it can be implemented in both for-profit and non-profit settings, for open-source innovation as well as for closed-source innovation, for collaborative purposes as well as for competitive purposes. Furthermore, it reveals that intellectual property rights are not really needed for successful drug discovery and development for the benefit of society. The operating principles of the core model are exquisitely detailed in two recent books. The first one, The World's Healthcare Crisis, from the laboratory bench to the patient's bedside, and the second, The Core Model, a collaborative paradigm for the pharmaceutical industry and global healthcare. Current efforts and alliances to develop a COVID-19 vaccine and/or therapy, including the key players, resources, experiments and strategies at an international level, are largely disorganized, uncoordinated, misaligned, and scattered, without coherence. The research and development activities and platforms, despite establishing important partnerships, are not in synchrony with one another, nor with individual national public health policies and socio-economic strategies, such as welfare, national security, economic stimulus packages, etc., all aimed at containing the pandemic and provide relief to their citizens. It is an urgent and critical need to have a centralized, open, and cooperative international COVID-19 initiative based on the core model in a strategic place, for example Europe or the United States, with multiple partners and key centers located in different parts of the world, from academia, the pharmaceutical industry, government agencies, such as the NIH and like institutions, consortia, pharmaceutical regulators, the United Nations, the European Union, governmental healthcare ministries, philanthropic organizations, and advocacy groups from all over the world, working together in real time, exchanging information and models, addressing problems, analyzing and synthesizing data, making decisions in a very organized and focused manner. This is how it was done by the Event Horizon Telescope Project, EHTP, in order to obtain the first photograph of a black hole, or the Human Genome Project, among many other collaborative initiatives. It is obligatory for the creation of a global strategy, an inventory of resources and assets, of potential partners, a list of haves and have-nots, using the most sophisticated platforms, including artificial intelligence, which is ideally suited for this endeavor. The operational leadership should be centralized for the core model to be effective. First, it needs to be decided whether the iCOVID-I initiative will be an open or closed cooperative system. In the discovery phase, the core model could be extremely instrumental because it could bring together a number of different partners from the public and private sectors with complementary technologies, learnings, expertise, and infrastructure together under one single and focused enterprise, the International COVID-19 Initiative. These partners could contribute with and exchange information on their basic knowledge of the structure and behavior of the virus and how it affects human cells and the human body. Based on this, a series of strategies, depending on the assets and strengths of each partner, can be formulated for the development of COVID-19 vaccines or drugs to treat the symptoms of the disease. The partners would freely exchange information, molecules, biological materials, animal models, and platforms in real time, and perform all the necessary preclinical testing until having a clinical candidate. Once the clinical candidates for vaccines or drugs are ready, and after reaching a consensus in the clinical development strategies, locations, recruitment and protocols, and involving the regulators, different Phase 1 safety studies would run in parallel. Using translational research principles, the same is applied for efficacy testing in Phase 2. At this point, the most successful investigational vaccines or drugs could receive accelerated review approval, or emergency use authorization, EUA, from regulators, and continue on to large-scale Phase 3 trials. For manufacturing and worldwide distribution, the parallel, not waiting for Phase 2 and 3 results, operational model set up by AstraZeneca in partnership with Coalition for Epidemic Preparedness Innovations, CEPI, Gavi, and the Serum Institute of India, the world's largest manufacturer of vaccines by volume, to mass produce up to 2 billion doses of AZD1222 vaccine, could provide a roadmap, an unprecedented, bold and gutsy, ready-to-go commitment if the vaccine checks out for safety. There are at present several high-profile collaborative efforts in motion and operational. They would be even more effective if they fully understood the core model, which is actually what they are trying to implement. For instance, ACTIVE, the initiative in which the NIH is partnering with other agencies in the Department of Health and Human Services, DHHS, including the FDA, CDCP, and Biomedical Advanced Research and Development Authority, BARDA. Other United States government departments, including the Department of Defense and Veterans Affairs, the European Medicines Agency, and representatives from academia, philanthropic organizations, more than 15 biopharmaceutical companies, and the Foundation for NIH 20 follow the core model very closely. Another example is one taken by Sanofi and GlaxoSmithKline, in which these otherwise rival companies are collaborating against COVID-19. Both companies will share their technology for this aim. Sanofi will contribute with its S-protein COVID-19 antigen based on recombinant DNA science, while GSK is contributing its adjuvant technology. Both firms are powerhouses of manufacturing, so their combined capacity will allow them to mass produce hundreds of millions of doses yearly. A third major effort is the alliance between Gilead, Novartis, Schrödinger, Takeda Pharmaceutical, and WuXi AppTec to share ideas, resources, and data with the goal of developing custom pan-coronavirus antivirals. It has been reported that the nature of this alliance is not commercial, but philanthropic, and that the tools that are discovered during these efforts will enter the public domain. The authors have clearly expressed and demonstrated that academia and industry and public-private partnerships and collaboration are not just nice things to have. They are essential for the creation of effective therapies. They are even more critical now in our fight against COVID-19. The implementation of the core model through the International COVID-19 Initiative, which incorporates a proven paradigm for efficient drug discovery and development, is our solution for the COVID-19 pandemic crisis. It is an unprecedented, centralized stewardship, fostering global collaboration to combat the global pandemic. It is a new way of thinking, and we must embrace it and execute it now. The International COVID-19 Initiative should also be a permanent organization, since we should expect similar or worse pandemics, related to SARS-CoV-2 or not, in the future. The lessons learned will guide us in how we deal with future pandemics. Conclusion: moral, ethical, and social responsibility. Our global readiness for a pandemic such as COVID-19 has been surprisingly poor, judging by the best estimates of at least 9 million people infected and 470,000 deaths in 213 countries, as mentioned earlier. The astronomical emotional and economic impact that we are experiencing today is only the tip of the iceberg. There are many unknown and unavoidable challenges ahead of us. A fundamental question we must ask, and for which we ought to have a convincing answer, is: what is the origin of SARS-CoV-2? At present, there are 3 main hypotheses about it. First, that it was genetically engineered at a Center for Emerging Infectious Diseases at the Wuhan Institute of Virology. Second, that it is a natural virus that was either intentionally or unintentionally released from the Wuhan Institute of Virology. And third, that the SARS-CoV-2 is a virus of natural origin with a subsequent zoonotic transmission, that is, from animals to humans, as was the case with Ebola, HIV, and SARS. To this day, the answer to this question remains uncertain. However, what is certain is the Chinese government's confession that the seafood market of Wuhan is the source of COVID-19. And inarguably certain is the fact that China allowed the spread of SARS-CoV-2 across the world by permitting international flights to and from Hubei province, the epicenter of the new coronavirus outbreak, while taking stronger measures to ensure that the virus did not spread within China by cutting off travel from Wuhan on January 23, 2020. Why did China allow international travel from Hubei to the rest of the world long past January 23rd? That question has to be answered, so another epidemic from China does not spread like fire throughout the world, ever again. A disclaimer regarding the content in this episode. The COVID-19 pandemic is a dynamic and rapidly evolving event. The numbers used in this episode correspond to data available as of June 22, 2020, when the final draft of this article was submitted. This article was written by Ibis Sanchez-Serrano and Rathnam Chaguturu. Ibis Sanchez-Serrano is a world-leading expert on healthcare. He is the president of the Core Model Corporation, Panama City, Panama, focused on global healthcare and biopharmaceutical innovation and translational science policy. He is the author of 2 landmark books on the world's healthcare crisis and the solutions to it, including the Core Model. He is a regular speaker at major healthcare-related conferences, international TV guest commentator, and an editorial board member of the scientific journal Gender and the Genome. Rathnam Chaguturu is the Innovation Tsar, founder and CEO of IDD Partners, Princeton Junction, New Jersey, United States, a non-profit think tank focused on pharmaceutical innovation, and was most recently Deputy Site Head, Center for Advanced Drug Research, SRI International. He is the founding president of the International Chemical Biology Society, a founding member of the Society for Biomolecular Sciences, and Editor-in-Chief Emeritus of the journal Combinatorial Chemistry and High Throughput Screening. If you've enjoyed this episode, then you can subscribe to Drug Discovery World free of charge by visiting our website at ddw-online.com, where you can also view all of our articles including references and images, and download the original PDFs. The links are in the show notes. If you've enjoyed the podcast, then do leave us a review and subscribe, and you can also follow us on LinkedIn and Twitter. Thank you for listening, and we'll hope to see you in our next episode.